Hello and welcome back to Psychology 300. This is Dr. Williams, and this is the lecture series over Chapter 11 in our textbook, Single Case Designs, Quasi-Experimental Designs, and Developmental Research. This lecture in the series, Chapter 11C, deals with single subject designs in general. Later we'll talk about each one individually. The learning objectives for this particular lecture involve you being able to explain when a single subject design would be preferred over statistical group design, and explaining how single subject designs demonstrate cause and effect relationships. Alright, so why would you ever use a single subject design? So much of our class is dedicated to group designs that it seems, by the time we get to this point, it seems as though the idea of using just one subject would be less than valid, that we would not be able to draw conclusions about treatment efficacy or the effects of an independent variable on behavior if we're only using a small number of subjects. But there are actually good reasons for using single subject designs, and they are as experimentally valid as a group design. They're just have different they serve different purposes. Single subject designs are primarily intended for intervention specialists or investigations that deal with working with real clients, where the focus is on the individual person and the treatment efficacy for that individual. A great deal of you plan to go on into psychology as a helping field, and you will not be doing research designed to assess the mean and the standard deviation of different groups, or the effects of an independent variable on the mean and standard deviation of a particular group. You'll be interested in whether or not the individual client that you're working with actually shows improvement based on the treatments that you decide to employ. And so single subject designs are really designed to document the functional relationship between a treatment variable and behavior for a particular individual. They are used to determine treatment efficacy and even across subjects, but the way we do this is to focus heavily on and intensely on individual subjects, a few individual subjects, and rather than to focus on a large group of subjects where you only collect one data point. Okay, so single subject designs typically are repeated across several subjects to demonstrate generalizability, but we never compare subjects to each other. Instead, we only compare each individual to their own individual baseline, and we draw conclusions based on that. The type of analyses that are used are called visual inspection analyses. They still rely on null hypothesis testing logic. There's a very sophisticated system for analyzing graphed data. They rarely use statistics, and when statistics are used, they're only used as secondary analyses, primarily because we're really looking for meaningful change rather than probabilistic change. All right. So single subject designs are used by intervention specialists and clinicians when they treat individual people in order to demonstrate that the treatment that they're using is working or not. It's used in a variety of areas. Applied behavior analysis is the primary area that it's used, but it's also used in special education, in school psychology, occupational and physical therapy. It's used in industrial and organizational psychology, and it's used in animal behavior, animal training, and animal husbandry. Single subject design is a valid and legitimate research method that uses each subject as a complete and intact experiment, and the use of multiple subjects is used to demonstrate generalizability. All right, so single subject designs do not require a lot of people or a lot of subjects. Often we only have 3 to 6 subjects, but for each subject a great deal of data is collected. For my dissertation, I only had 6 birds, but I collected in excess of 100,000 data points. So, and only across those 6 birds. Group designs collect a lot of data, but they do so by taking a snapshot of each subject, usually just once, or several times in a within subject experiment, but each condition is represented by only a single data point per person. Here, each person is a complete experiment in and of themselves. No additional people are required. However, the use of 3 to 5 or 6 people are often employed to demonstrate generalizability. In order to demonstrate change, the criterion for change is based on replication. Single subject designs use fewer subjects, and they study them in depth across multiple treatment conditions and, importantly, across replications. Conditions are not only administered to every subject, but they're often administered more than once in order to demonstrate reliable effects of the presence or absence of the treatment intervention. The therapeutic criteria is used in addition to an experimental criteria. So simply saying you want p less than 0.05 is to say that there's a probabilistic statistical change. But within single subject design, we often are looking for therapeutic changes. So changes that are not only significant at a statistical level, but more importantly, that make important, meaningful change in the client's life. Reliance on replication within individuals and across individuals is how we demonstrate cause and effect. And how we rule out individual differences is to have participants serve as their own controls. Experimental criteria for demonstrating cause and effect relationships between an independent and dependent variable are usually detected using statistics in group designs. Here, in the single subject design, we demonstrate this experimental control by looking at stability within each condition. So we'd like to see that a client in any condition, whether it's a control baseline condition or whether it's a treatment condition, we usually wait until their behavior stabilizes. And we use that stable data to demonstrate performance in a condition, and then differences between baseline and treatment conditions are then used to show cause and effect. Replications provide evidence of consistency of treatment effects across individuals. So by replicating across people, we show that it works for one person, another person, another person. And it's stability within each condition and replications within each person that demonstrates our treatment effect. The therapeutic criteria, however, for treatment efficacy is not based on statistical change, but rather meaningful functional change that makes a difference in someone's life. So social validity is extremely important in single subject design, and we often employ subjective evaluation by consulting with significant others. So we take our data and we sit down with parents or teachers or employers and ask not only are the differences real, but are the differences meaningful? Let me give you an example. If I were studying self-injury in a group of children who engage in self-injury, and I have a treatment that I would like to try that should reduce self-injurious behavior. So I gather data carefully on baseline rates of self-injury, and then I employ my treatment, and I see whether or not that self-injury goes down. If self-injury is quite high at baseline and it's reduced by 50%, that might be statistically significant. But for applied behavior analysis, for applied psychology, 50% may not be sufficient. And so we would not necessarily call that treatment effective, even if it cuts the behavior in half. If the degree of self-injury is still life-threatening, even at lower levels, it's not considered an effective treatment. So in this case, we're now looking at not just experimental criteria of demonstrating cause and effect, but also a therapeutic criteria for treatment efficacy to determine whether or not a treatment makes a meaningful change, not a significant change, but a meaningful change in the behavior of interest. So the philosophy of single subject design, which is important, is that we treat participants not as participants in an experiment, but rather as individuals or individual clients. And we don't assess treatment efficacy according to some mean and standard deviation of some manipulation, and we don't determine, we don't use techniques that are the result of a study of a group of people by someone else, and where the statistical significance is based on the mean and standard deviation. So let's say, let me do it this way. I don't pluck a treatment out of the literature and just apply it and assume that it will work. Instead, we rely on the clarity of raw data, on the robustness of the effects that we see for that individual, and not on probability theory, and not necessarily on statistics. We are very much less concerned with p less than 0.05 alpha levels, and single subject design avoids all the flawed assumptions of statistical designs, of which I haven't had time to really go into for you, but perhaps at some point I'll be able to outline those for you, because I think it's important to recognize that all approaches have their flaws. What is critically important to this kind of research is that we have a stable baseline so that we know what the behavior looks like in a stable form before we begin with our treatment, and that when we implement our treatment, we again wait for stability. And in many cases, we're talking about, or at least in the case of ABA reversal designs, the replication of treatment and baseline, the alternation between treatment and baseline, going back and forth between treatment and baseline, putting the treatment in, taking it out, putting it back in again, taking it out, and watching behavior track those changes is what allows us to determine the cause and effect functional relationship between treatment and behavior change. All right, so next we'll move on to the specific designs.